Hello, my name is Melanie, and today I'm discussing the article Blue Light Exposure in Vitro Causes Toxicity to Trigeminal Neurons and Glia Through Increased Superoxide and Hydrogen Peroxide Generation. Both natural and artificial light has been recognized as playing a role in various ocular diseases, and blue light spectrum is exceptionally emitted by artificial sources. This has been increasingly implicated in the topic of photosensitivity and often associated with ocular pain. This neuropathic signal is sent from nociceptive trigeminal ganglia, which further travel through the ophthalmic nerve and its distal branches. The goals of this study were to investigate cytotoxic impact of wavelengths of visible light on primary cell culture of mouse trigeminal neural and glial cells, asking if blue light has an impact on these non-visual nociceptive neurons, and delving into how these light signals are received and the pathways involved in this phototoxic message. Trigeminal ganglia cultures from 10 adult male mice were split into no treatment or antimitotic treatment group. The antimitotic treatment, Ara-C, was employed in the study to determine if phototoxicity would depend on cell population. The cells were exposed to 5 various wavelengths of light with a dark control. Light at 410, 440 nanometer wavelengths is known to be harmful for the retina. The 480 nanometer wavelength is implicated in circadian rhythms. The 510 nanometer wavelength of light is reported to be soothing for photophobic migraineurs, and light at 630 nanometer wavelengths was used as a second control as there are no current data reporting damage from red light exposure. Researchers used cell titer glow and ROS glow H2O2 assays. Cells were stained using Yopro, propydium iodide, and DAPI, as well as Mitosox red mitochondrial superoxide indicator dye, Mitotracker deep red FM dye, as well as the following antibodies. Both melanopsin and neuropsin are light-sensitive photopigments that play an important role in non-visual functions such as circadian photoentrainment, but precise functions of neuropsin are still unclear, so anti-melanopsin and neuropsin stainings were employed to indicate the presence of these non-visual opsins. The 410 nanometer wavelength was found to significantly decrease cell viability in both groups, and additionally in the Ara-C group at the 440 nanometer wavelengths. DAPI, Yopro, and PI fluorescent stains, all used as indicators of cell death, showed significant increase when exposed to the 410, 440, and 480 nanometer light. At 410 nanometer wavelengths, all 3 stains were visible, indicating that these cells exhibited both necrotic and apoptotic processes. GAD153 antibody staining revealed both growth arrest as well as DNA damage following illumination of the no Ara-C group at 410 nanometer wavelengths, and in the Ara-C group at wavelengths of 410, 440, and 480 nanometers. Under the 410 nanometer light condition, anti-pan staining revealed decreased numbers of cell bodies and indications of neuronal damage. Measuring markers of oxidative stress, a significant increase was found in both hydrogen peroxidase and superoxide anion levels at the 410 nanometer wavelengths for both conditions, but the Ara-C group showed increased rates of hydrogen peroxidase additionally at 440 and 480 nanometers. Corresponding fluorescent signals from superoxide anions belong to glial cells instead of neuronal cells. Biomarkers implicated in neuronal activation and cellular inflammation were next analyzed, revealing 410 nanometer light to significantly increase ATF3 rates and rates of CFOS and interleukin 6 in both groups. Levels of CCL2 were found to be significantly increased in the Ara-C group for both the 410 nanometers and the 480 nanometer wavelengths. Using the PERK1 and 2 antibody staining, both neurons and glial cells showed a decrease or even complete absence of fluorescent signals for both conditions. Among glial cells, an increase of fluorescence following 410 nanometer light exposure corresponded to compromised mitochondria. Immunostaining with anti-melanin antibodies revealed immunoreactivity in neurons for all light conditions and for the control. Anti-neuropsin staining was detected in glial cells for all wavelengths of light, but detected in neural cells only after exposure to 410, 480, and 630 nanometer wavelengths. This phototoxic light through respiratory chain and neuropsin is absorbed by glial cells, thus increasing oxidative stress and glial cell death. This chain reaction reaches neurons and damages them via the ER pathways with additional oxidative stress and nociceptive message generation. By assessing different wavelengths of blue light, researchers were able to gain a better understanding of the phototoxicity of these wavelengths, but also to the precise mechanisms underlying the common symptoms of increased sensitivity and ocular pain in given illumination conditions. Understanding these mechanisms can perhaps widen the scope of treatment and prevention. But something you can do today is take a break from screens. You've earned it, and your trigeminal neurons will thank you. Thanks for watching, and if you have any questions, feel free to reach out.